Hi Nat5, so we are looking at food production still in the life and earth topic. Right, so we've looked at how you might be able to increase the yield of a plant, but there is another way you could increase yield, and that's to stop damage that's being done by other things. Okay, so a pesticide is any chemical that kills pests. Okay, so and a pest is anything you don't want on your crop or on an animal. Okay. And you can name the pesticide a little bit more specifically because you could decide on a type of pest you want to target. So we could actually say herbicides if we wanted to kill plants, fungicides to kill fungus, insecticides, you get where we're going with this. Okay, and you decide on how you apply them depending on what it is and how much you're treating. So you might go with the little spray bottle, the big spray bottle, a much larger spray, or a ridiculously huge spray. So pesticides are really important that they're understood because there are lots of properties that are important about it. So here's your first one, toxicity. So what that means is basically how much of that you need to kill something. If you're interested, go look up a concept called the LD50. It's called the lethal dose 50, which there is for every single chemical pretty much on the planet. It's interesting, although quite worrying as a thing. So basically, the more toxic it is, the more precautions you would need to use when using it, and you'd have to be very aware of that. And then we've got another one called persistence. So persistence is how long it stays in the original form you put it on. So if you spray with this particular chemical, how long is that chemical there for? And another kind of extra thing that you could look up is something called biotransformation, which is quite interesting about what can happen with certain chemicals. So quite a lot of chemicals break down naturally. That would be biodegradable ones, but some don't. So you may have ones that can hang about for quite some time. So the problem with having a persistent chemical is that you get two kinds of things that can happen. So one of the first ones is called bioaccumulation. And what we mean by accumulation is that if it doesn't break down, then that just means that it'll keep building up. It's kind of obvious. So depending on the chemical and its toxicity level, you might actually manage to build up enough toxin to kill something over their lifespan. So they start off with nothing and then it gradually builds up and reaches a toxic level, and that's it. So this specific term, don't worry about too much. The next one you definitely do. So what is even more important with persistence is if you think about your pyramid of numbers, okay, so you start off with a toxin. If it doesn't break down, then everything from the bottom of the pyramid will end up in the top of the pyramid. So the ones from above end up with all of the chemicals from the one below, and it gets more and more concentrated then as it goes up the food chain. So there's lots of examples of where this has been really quite damaging. So there's a place in Japan called Minamata. And what happened there was you had lots of people who suddenly started getting mercury poisoning, okay, and it was called Minamata disease in some places. So what they found out eventually was that what was happening was in a mine, they were using mercury as part of the mining, and so the mercury basically was getting into the water. And then you had, mercury doesn't break down, so you have little amounts in the kind of algae and then the small fish. And then in the next fish up, you had quite a few, and now you've got more. And eventually in the biggest fish that you had really high levels, and then the fish was getting caught by fishermen who were then eating the fish, and the concentration had now got so bad that it was causing real problems. So one of the other really good examples we have of this is something called DDT. I'm not even going to attempt to say that one out loud. Okay, now DDT is an insecticide, and it's a really good insecticide, and it was used as an attempt to fight against malaria. So what they were trying to do was to knock out the mosquito transfer bit, and it was also used as an agricultural pesticide. So the first problem here was biomagnification. So what was happening was in the water, if we look here, we've got 0.000003 parts per million. That's a very, very low amount. But then in the small animals, they were building up to 0.04. So if you had several of these small animals were getting eaten by the small fish. All of the stuff that was inside the small animals ended up in the fish. Then the large fish eats several small fish, gets all of the stuff. So in this case, we would say, you know, maybe they've eaten 4 of these small fish. And then the large fish is getting eaten by the fish-eating birds of prey, much, much higher level. And if you look at that magnification, that's huge, absolutely massive, because that's gone from 0.000, we're going 1, 2, 3, 4, 5, 4, 5, 3, up to 25. You know, so just just moving this along, 1, 2, 3, 4, 5, 6 orders of magnitude. So it's a real big problem. Okay, so the second problem with DDT was actually something called resistance, which we're going to look at in a little bit more detail in the key area on evolution, but I think it's worth putting in here so we've got a context. So we used it to try and remove mosquitoes because this is a problem. It's a huge number of people are affected by this. So you sprayed it on water and trying to kill off all the larval stages of mosquitoes. And that was really quite effective. But what they found was that there was a resistance recorded in 1959. So what they found was that there was an area where they'd sprayed, but it didn't kill all of the mosquitoes. And so what had happened was you had a mutant that was immune to the DDT. And what you've now done for that particular mutant is wipe out all of the competition that it had. And so it did really well and passed on that genetic mutation, which meant that the next generation were a lot less likely to be killed. So we had a kind of decreasing mortality quite drastically once this resistance appeared. So what are your alternatives? And this is what you need to know as well. So we could use biological control, and that is the term that you need to use, biological control. And what we mean by that is a particular organism is used, a living organism, to try and control that particular pest. So the type of organism we can try and bring in, we could bring in a predator, we could bring in a parasite, or we could bring in a pathogen. Okay, so a disease form. So have a look. We have a cabbage looper being killed by Bacillus I can't even say that one, thuringiensis. So that's a pathogen. We have, here's an egg of a corn earworm being attacked by a trichonogama, so this is a parasite. We've got aphids being eaten by ladybirds, that's a predator. Here we've got disease, attempting to wipe out rabbits with myxomatosis. A particularly gross picture of wasp larvae coming out of a caterpillar, it's a parasite. We've got rove beetles attacking a maggot, so it would be predators. Another one with wasps, pretty gross. And another pathogen that is destroying the larvae of this particular nematode. So pros and cons. Compared to an out-and-out chemical, you would say that there's less danger of impact to the environment, but one of the cons might actually argue against that. It's less likely to impact on water quality. Certainly, you're less likely to put in a pollutant that is going to magnify or anything like that. You're not getting bioaccumulation, biomagnification. You've got none of these issues, but can be really quite high cost. Might take longer, so you might actually overall lose more than you wanted to. Could be quite time consuming to maintain, and quite often you have a potential risk here. This is this less danger thing argument, because it could impact on other food webs, other parts of a food web, because you might have a predator that you introduce to get rid of an insect that then feeds on other insects that were important to, say, pollination of another plant. You know, there's all of that potential impact. So quite often biological control, to try and actually do it well and maintain it well and look after it, you try and do it in enclosed spaces, so like your polytunnels, greenhouses, things like that is one of the maybe the best places to use this. And that's us.